The genome is incredibly complex. There's 3 billion base pairs, and we desperately needed tools to unravel the function of our genomes and how they regulate the expression of genes. With that understanding, we can really transform how diagnostics are made in the clinic, but also how therapies are nominated and proposed for various diseases. I am Jason Buenrostro, and I'm a cellular and molecular biologist. In order to understand how a genome relates to how our cells work and therefore how tissues and organs work, we have to understand how genomes are being regulated and how genomes are deciding to activate genes. That 3 billion base pairs of DNA needs to be folded into a tiny, tiny cell. So in order to understand how cells are working and how genomes are regulated, we need to understand what parts of the genomes are made open for the cell to use. The method of chromatin accessibility measurements is meant to directly address these opened versus closed fractions of the genome and to measure them across our different cells. So the ultimate goal of our research is to understand how the changes in genome structure and chromatin accessibility leads to changes in the expression of genes. In that process, we've discovered gene regulatory control across many different kinds of human cells. And now we are also able to measure that at single cell resolution to say, for any given one single cell, what are their opened and closed regions, giving us a unique opportunity to categorize and catalog cells by these configurations of opened and closed DNA. We've invented a method called ShareSeq, which allows us to pair the measurements between open and closed DNA and gene expression, and that, when applied to single cells, can give us tremendous insights into the mechanisms, the how of how genes are activated or repressed. We've been excited about seeing the maturation of our tools. It's already made a big impact in understanding the basic mechanisms of how diseases arise. I'm very excited about the opportunity for these tools to be translated to diagnostics, to really be able to make these technologies useful to inform clinical decisions of how diseases might be treated.